So first we have an announcement for all classes, although really this doesn't pertain to you guys so much because you're not my late crew, but I'm still going to say it. At the beginning of the year, I talked about my late policy, and it was just going to be 50% if you turn something in late, and then come test day, you don't get to turn it in anymore. And a couple of things. One, that leaves me looking through weeks and weeks of back information to see if there's somebody who turned something in and I didn't know it, and now I have to record it. And I literally spent 15 hours this weekend not doing anything but late work. 5 hours Friday, Saturday, Sunday, no creation, no moving forward, no grading of current stuff, which is really hard because your stuff, AP stuff, takes me a while to grade and intimidating. And like you guys, it'll get like intimidating stuff gets set aside. And I feel like I always have to catch up on all my late stuff before I can start something. Anywho, so that makes it, instead of 15 hours of grading that I could have gotten a couple of your things done, I was doing late work. So I'm altering it a little bit. In class, if you've had me, you know that I have a 10% rule. So I've always been 10% a day. The science department was trying to be common, and so we did 50%, but I'm going back to my way of doing it, which is 10% a day, which is beneficial to people who turn in late work, because if you had a 40-point assignment and you turn it in the next day, you got 20, and that was a hard hit. So this way you'll get 36 instead of 20. So it'll be 10% a day. And if you do the math, after 9 days, there's no point left. So after 9 days, I'm not accepting any late work. So I could harp a little bit longer, but like I said, this group is, you guys always get your stuff in, so it's not so much pertaining to you. But I want to put it out there so you know it in case that does happen. The second big thing on our list, are you planning to take the AP test? So we have a deadline of October 30th to sign up for the AP test. A handful of you, literally 5, have already signed up. Kudos to you, because Miss Snook hasn't pointed you in that direction. But if you go to the counseling page, advanced placement right here, registration, the cost is $99 per exam. So if you're taking multiple exams or not. We are not doing payment through the school, which we've done in the past. So we're outsourcing a registration site. So we're going to show that. If you happen to be on free and reduced lunch, you're just paying $5 for the AP exam by November 2nd. If you need financial assistance, send Mr. Lepper an email. And when you do, there's a 2-step process. So registration is one, and it's a little bit confusing because maybe in your AP Central site right now, it says you're registered for the AP exam because everybody was automatically registered. But after the 30th, if you haven't registered on your own and paid your money, your name will be removed from that registration list. So it'll no longer be on your AP Central. So don't be confused by AP Central if it says already that you've registered and you know you haven't. So you have to register, then you get a join code, and then you have to go through AP Central and input your join code, yada, yada. So then that's open till the 30th of October. And then after that, there's a $40 late fee. So times are changing. You might look at what the rest of the school year is going to look like. We're having some change. We're going to have 45-minute classes. I'm only going to see you once a week. And you're going to do a lot of on your own stuff. So when you're making your decisions on whether or not you're going to take the exam, I'm not telling anybody to not take the exam. I don't mean that. But I do think you should look at the schedule and see, are you going to be able to handle it? Okay. I'm going to give you all the information you need. It's just you're going to do more of it on your own. That's all. And that's really more of somebody else's rant than it is my rant. So I thought I would throw it out there to be fair. I had one more site for you. Okay, so if you're going to register, I can put this link in Google Classroom. It's also on the AP site. So this is the website. It's total registration is what it's called. See up here, total registration, AP. Here's a number, 231905, that's us. So you'll go through this, name, and that's that. Okay. Do you have any questions? You can chat it or you can shout it out. I love hearing your voices, so never feel like you can't speak up in my class. Okay, so we've gone through that process. So next up, we're gonna have a test next week. It's gonna cover chapters 4 and 5. Today we're finishing up chapter 4, and then we're gonna do chapter 5. Chapter 5 is cell membranes and diffusion, and a little bit more like active transport and stuff like that. We're gonna go into more detail with osmosis, diffusion, probably the following week, because we get to talk about math when we do that. So that's going to be next week. And we have, you know, Monday, Wednesday, and Thursday. So here's 2 thoughts on our test to get the most content time. I feel like I don't have time to give you guys any tests. I just want to do content all year and never test because I don't know where and I have time. When I only see you 2 or maybe 3 times a week. This week, once a week. So anyways, we can do it Thursday and have it maybe during the flex time so we still have content time. Or, I don't know, as in, is it feasible and am I allowed because it's not my day? Could we do it end of day Friday? What are your thoughts on that? That way you still get your 3 days of contents. And then, anyways, maybe I'll throw it in as a question in the Google Classroom and you guys can let me know what you're thinking. This class, probably more than any other class, I kind of let you drive me sometimes. Another question I had on the lab materials. The other day I had said, you have your choice. You can either do potatoes and iodine or you can do beets and bleach. And I'm wondering if you did go out and get those materials and you're super anxious to do that lab, or if you didn't go out and get those materials, then we don't necessarily have to. Oh, Ahmed, I think it went out in the email. I think you guys got an email yesterday. So, okay, yeah, so did you guys go out and get materials or were you kind of waiting for me to push you a little bit more? Because we don't necessarily have to do that lab. The more I thought about it, I was like, I did it with my freshmen. Do I really need to do it with my advanced kids? And you're going to watch a video today that kind of does the lab anyways. So if you didn't go out and spend your hard-earned money on that material and you didn't have your heart set on it, I thought, well, you'd just go ahead and skip that. Anybody feeling bad about that? Am I with you?
Yeah.
I mean, it's a little difficult, or not difficult, but more, I think, tedious to go out and get certain materials I don't have on hand. And also, when it comes to labs and stuff, I get very nervous. I'm going to mess something up and record the wrong data if I don't have, like, someone else, like, working next to me or, like, a teacher assistant with that stuff. That's just me.
No, that's fair. Thanks. Yeah, I didn't, because I did tell you to go get stuff, and so then I hated to retract it if you did go shopping. But if you didn't go shopping, then I'm retracting it, and we're good to go moving forward. Yes? This video does the bleach and beets, so you're going to get to see it anyways. Okay, so then today we're going to get through all things cells. After that, you have one video to watch and an accompanying slide set. So you'll want to probably open the slide set while the video is playing. so that you can answer some questions along the way. And then our lecture is going to end with endosymbiotic theory. So I'm just giving you one sheet to read and answer. It's like maybe 5 questions. Okay. And if you're interested, I can show you that hybrid schedule when we get done with our lecture, if you'd like. Okay, so moving forward, here we go. So a lot of this I think you're already familiar with. I really only spend one day on cells when we're in class. So this is our one day. So we know that all cells are surrounded by a plasma membrane, no matter what type of cell it is. We know that they all have cytoplasm, but we're going to start calling it cytosol. It's the same stuff, just in 9th grade you called it cytoplasm, and as we get going into higher levels, they refer to it as cytosol. So just so you know, they're the same thing. The cytoplasm is referring to the liquid portion and the organelles, to be technical. We know that we have genetic material of some sort, chromosomes, in the form of DNA. And then we have ribosomes that make proteins. So that's probably going to be our 2 most important structures, along with mitochondria and chloroplasts. So we know organelles means tiny organs, right? Okay, so looking at the prokaryote and the eukaryote, you should be able to compare and contrast, which means find the similarities and the differences. So when they say compare, we're looking at similarities, and contrast, we're looking at differences. So right away we can see a big difference is that the prokaryote has no nucleus and the eukaryote does have a nucleus. The prokaryote does not have any organelles, whereas the eukaryote does. They both have a cell membrane. They both have genetic material. They both have that cytosol, and they both have ribosomes. So you might not have recognized before that a prokaryotic cell has ribosomes, but it does. Pro is before, true. Karyo is referring to the kernel, so the nucleus. So before the nucleus, and true nucleus. And then we want to be able to compare and contrast plant and animal cells. So lots of structures, they have the same. The biggest difference is that plant cell has that central vacuole, which it uses to maintain the turgidity, the pressure against the walls, which is what allows it to be stiff, or if it doesn't have water, it falls over. And we know that the plant cell has chloroplasts and the animal cell doesn't. They both have mitochondria, so not to be confused with that when we talk about respiration. Lysosomes are another thing that's more common in an animal cell than a plant cell. Lysosome is for digestion. And then another thing that's more common in the animal cell is the centrioles when it comes to mitosis. Okay, so here's our comparison. Nucleoid region. So the nucleus is still in a generic region, right? A general region. It's just not contained within a membrane for the prokaryote. So they still have that area where the nucleic acid will be found. They have cell walls, all of them, whereas eukaryotes, we know only some have cell walls, right? So we know that our nucleus is enclosed within a membrane, the nuclear membrane, or the nuclear envelope, it might be called. And then we have cell walls that are in fungi made of chitin, cell walls in plants made of cellulose. We're clearly more complex because we have all that inner compartment space that divides and conquers all the jobs that a cell needs to do. So this, I'm sure you're familiar with the prokaryotic cell. Typically we see flagella for movement. We already talked about the nucleoids. I think the ribosome is the one that might be new to you because we would have talked about it having no organelles. And technically the ribosome isn't an organelle, right? So they do have ribosomes because all living things need to make proteins because proteins control all of our cellular functions. So the organelles, specialized structures. We know probably these over here on the right. We maybe didn't talk a lot about cilia and flagella, but they're for movement of one form or another, either moving the entire cell, or it might be like on protists, they have like little cilia that push food towards a gullet into an opening. Your cells have cilia to move an egg through the reproductive tract, to move dust and bacteria and whatnot through your respiratory system. So it's for locomotion in one form or another. So all these organelles, they're all contained within membranes that are continuous with or made of the same material as your cell membrane. So it partitions the compartments, makes spaces for specific reactions to occur. So it separates pH, salinity, concentration of different proteins, minerals, salts, ions, things like that. Okay, so that gives every space the perfect environment for the reactions to occur, because we know all of our enzymes are very specific to pH and salinity and such. Then membranes as sites for chemical reactions. So we want to get used to the cell membrane is where reactivity can occur. So you'll see a lot of folding, and that's one of the things you're going to look at in the video today. Inward folds increase surface area. So if you've had me for anatomy, we talked about that with the brain and the stomach. Like our brain, if you look, it has all these inner foldings. Our stomach has the same, rugae, folds on the inside. Your intestines have little villi, which is folding of the membrane, and that's to increase surface area. The mitochondria has all that inner folding, the cristae, right? And we know we have the grana. All of that is inner foldings of the membrane. So you're increasing surface area so more reactions can occur. Okay, so basically, what do we need for cells to live? We need proteins and energy. And remember, we wanted to be able to explain why we need these things, not just we need them to stay alive, but be very specific scientifically, what do we use them for, right? So proteins are used for every cell function, moving material into and out of the cell with active transport, as enzymes taking part in all of the chemical reactions, sending signals from outside the cell to inside the cell. They have lots of jobs. Energy we need for cell growth, cell reproduction, respiration. We talked about free energy and how every reaction has a certain amount of activation energy that they need. So for all of our chemical reactions, we need to overcome that. So growth, repair, and renewal. So some of our cells, like we're familiar with the repair portion because we all get little scars. But all living things go through that, right? So they need them to make more cells so the whole organism can get bigger, or unicellular, so that they can reproduce and create more of themselves. So building proteins, we want to focus on the central dogma, right? So we go from DNA to RNA to proteins. So we know that DNA is contained within the nucleus. It needs a way to get from the nucleus to the ribosomes. So it goes into an mRNA form that is small enough to escape the nucleus, get to the ribosomes. This is where the proteins are made. Then what happens to them? Then they're shuttled off into the ER. Remember the ER is the endoplasmic reticulum, which is used for transport. So the ribosomes that are either free-floating or embedded on the endoplasmic reticulum will be shuttled off to the Golgi Apparatus. What happens here? It's the packaging factory. So it packages and modifies proteins. So different proteins need to be that final folding, right? So they need some modification. They might need methylation or acetylation. They might need some sort of compound added to them before they're ready for export. So they're packaged for export, and then the packages are vesicles. The vesicles make their way to the cell membrane. They have the same outer membrane as the cell membrane, so they can fuse directly with the membrane and open up and release their contents. So in the next chapter, chapter 5, we'll talk about that exocytosis and where that material will be released. So you should be able to talk through this whole assembly line process. Any questions so far? Okay, so ribosomes, we can go into a little, oh, I hate when my font does this. We can go into a little bit more detail in that they're made of 2 subunits, the large and the small. And this is one of the things that we can use to compare prokaryotes and eukaryotes, because their structural units are slightly different. Like the size of the units, or the, like when, I want to say precipitate, but that's not quite right. Anyways, the size is different, and that's one of the things that we can use to compare prokaryotes and eukaryotes, and also to support the idea that mitochondria and chloroplasts were once bacteria, because their ribosomal subunits are of the same kind, of the same size as the bacteria, and not the same as the rest of our cells. Okay, so we know ribosomes make proteins. What are they made of themselves? They are made of proteins and rRNA, ribosomal RNA. Okay, so they kind of have an enzymatic function in that they're holding things together so that the chemical reaction can occur. So you can see the rough ER and the smooth ER. The rough ER has the ribosomes attached, associated with protein synthesis. The smooth ER more associated with detoxification and lipid synthesis. And this is what it looks like under a microscope. Okay, so we can see the inner foldings here, right? Everything is like one continuous membrane, and it's just inner foldings. So this is the cristae. Oh, no, it's not. I didn't read that right. I was like, oh, I guess I'm looking at a mitochondria. So this is your ribosome right here. And this is the mRNA feeding through it. So tRNA would bring our polypeptide, our individual amino acids, which are binding together to create a polypeptide. So you've seen that, and we'll spend more time on that another day. So the rough ER, protein secretion. So secretion versus excretion. So excretion is getting rid of waste. Secretion, you're exporting proteins that the body needs or other cells need for some form or another, right? So it could be a hormone, and you've packaged up a hormone and you secrete it to be used for other purposes. So when you think excretion, you're thinking that you're getting rid of it for good, it's waste, but secretion is a purposeful transport. So they're packaged into transport vesicles, which we can see here kind of plopping off of the Golgi apparatus. They make their way and fuse with the cell membrane and then open up and release their contents to the outside. So the Golgi is for modifying and packaging. You can think of it that way. And we want to get used to calling these vesicles. So secretory vesicles will have something to do. This is just that whole going from the nucleus. We can see how the ribosome or the information can get out of the nucleus through these nuclear pores. Makes its way to its ribosomes, which are very, very close, so it doesn't have to travel far. Ribosomes make the proteins. They travel through the ER, are released to the Golgi apparatus, changed in some form or another to take on their final functional shape, and then they are sent out through the vesicles for export. It's a lot of reiteration, really. So the smooth ER is a little bit different since it doesn't have the ribosomes attached. It's not associated with that protein synthesis, but you can associate it with lipids. So it synthesizes lipids like oils, phospholipids for the membrane, steroids, which are signals sent from one cell to another, and sex hormones. Then hydrolysis. So it's going to hydrolyze glycogen. So glycogen, remember, is the animal form of starch, basically. It's our glucose polysaccharide stored in the liver and muscles. And we break it down to release energy, to release individual glucose molecules into the sugar, or into the bloodstream. so their blood sugars. And it also detoxifies drugs. So again, that's going to be the job of the liver. So you would expect the liver to have lots of this smooth ER. Lysosomes, remember lice is digesting or breaking apart. So we know the lysosome, some is body. So it's a digesting body. So the digestive system, the stomach, the garbage disposal, you'll hear lots of different analogies for it. But basically it's going to break down molecules. And it could be that it's old, worn out cells or parts of cells, or in the digestive tract. Clearly it's going to have to do with breaking down food. But for a cell itself, like endocytosis, we've talked about how the cell brings stuff in. It's going to fuse with these digestive enzymes, be surrounded by a vesicle, and then that becomes a lysosome. So now it will be a digestive body. So they're going to fuse with food vacuoles. Here's that picture that I was just talking about coming in, endocytosis. Notice the membrane surrounds it. It's going to fuse with other enzymes. Here's the enzymes. And then the lysosome will be able to digest that coming from the Golgi, making that connection, right? Because they packaged those proteins. Then it'll break down what the cell brought in. So like white blood cells in our body would have a lot of lysosomes because they have to break down all of the foreign bodies that they're ingesting. So some of our cells are going to naturally be programmed to die off. Lysosomes can be used to kill cells when they're supposed to be destroyed. And you might be thinking apoptosis when you hear that, programmed cell death, right? So like the tadpole originally has a tail. Where does the tail go? It doesn't just drop off. We say that it's reabsorbed, but what does that mean? So these cells are actually digesting that material. So the lysosomes are in charge of morphine, changing form. You can also see it in the human bodies. That's on another slide. So this is how the tail gets rid of it. It's how we get rid of all the webbing between our fingers. Some of us haven't. Some of us have that webbed hand. I forget the dactyly word. Anyways, so you can see here, you start out with, you know, a paddle basically, and how do we become individual fingers? That would be the job of the lysosomes through apoptosis. So all of our form, like originally we're just a big blob, and how do we get arms and legs? We have to get rid of the cells that are embedded. There we go, syndactyly. I should have known that one, Okay. Then we're getting into the mitochondria and chloroplasts, which we spend a good amount of time on in chapter 8, talking about cellular respiration and photosynthesis. So we know the mitochondria is the mighty mitochondria. It makes energy, right? So it's going to make ATP from the breakdown of, I'm going to be very generic here and say glucose. But when we get into that chapter, we're going to learn how we actually break down lipids, proteins, and nucleic acids in order to release those energies. Chloroplast does the opposite, right? It takes the sun's energy and stores it in the form of the carbohydrates. Okay, so the big thing about these guys is making energy in one form or another, transferring energy from one form to another. They both have double membrane systems, and this is what I was pointing out before with that inner folding. So the inner folding causes or allows them to have more reactivity because they have a greater surface area. They both have internal ribosomes, DNA, and enzymes. So they function independently of the rest of your cells. They reproduce independently of the rest of your cells. They have their own genetic material, which is not the same as the rest of your cells. Okay, so this is your mitochondria. We should be familiar with the greater structure of the mitochondria, the outer membrane, the inner membrane. So the inner membrane we call the cristae, all those inner folds. The intermembrane space is in between the outer and the inner membrane, and in the middle of it all, inside of all of the cristae is your matrix. Okay, so we're going to talk about hydrogen moving from one side to the other side. So we need to be able to talk about those places in the mitochondria. So we know cells that are going to have a lot of mitochondria need a lot of energy, right? Not all of our cells have the same contents. So muscle cells, they would have a lot of mitochondria because they need a lot of energy. Interesting thing about the mitochondria, your mitochondria have only the genetic information from your mom, none from your dad, because when the sperm enters the egg, remember only the head enters and the mitochondria is in the body. So dad's mitochondria doesn't go into the egg, so your genetic material that's in your mitochondria is only from your mom. You can remember that on Mother's Day, okay? Say thanks for the mitochondria, Mom. So the chloroplast, a little bit different. We have different type of plasts. Yes, Cameroon. What can I do for you?
So this is more just out of a question. So we have only the mitochondria from our mothers. Does that mean they have their mothers and the mothers before them? And so would that mean I have like my great grandparents' mitochondria?
Yes, the mitochondrial Eve.
Oh, that's what we think about.
Yep, all the mitochondria are like can be traced through the females of your family, of your mom's side. And so they refer to the mitochondrial Eve because supposedly we can trace it all the way back.
Huh. That's cool. All right, that was all. I just was curious.
So remember that on Mother's Day.
I will.
All right. So plants have plastids. We probably are most familiar with the chloroplasts for the green, chloro green, but they have other plasts that give them different colors, right? Like you see that in the flowers and you see it in the leaves turning color now. So chromoplasts, they just store pigments. Pigments, remember, reflect a certain wavelength of light, and that's what gives it the color. So we see that in our fruits and our flowers. Amyloplasts, that's where we store our starch, and so they're in greater concentration in the roots and in tubers, which are similar to roots, big fat engorged spaces underground. And then the chloroplasts, again, we wanted to be familiar with the inner foldings, which are the thylakoid membrane. And remember, individual stacks of thylakoid put together to make a grana, a big stack. Because we'll talk about that even more. So that's where we get the green. So organelle is not part of the endomembrane system. So before I was showing you how that membrane was consistent between the nucleus and the ER and the Golgi all the way out to the plasma membrane. So these, as I said, are independent of the rest of your cell. So they grow and they reproduce on their own. They have their own ribosomes for making proteins. So that means they have their own chemical reactions occurring. They have their own circular chromosomes. So one of the differences between the bacteria and us is that we have that chromosomes that are linear in shape, but and we have lots of them, and different animals have different, or every organism has a different number of chromosomes, but bacteria just have one chromosome and it's in a circle. So mitochondria and chloroplasts both have one DNA, one genetic material, and it's in a circle. So again, that's another similarity between those cells or those structures and bacteria. Okay, so structurally, they're very much like a bacteria, which leads us into a hypothesis that they came from bacteria to begin with. Okay, so who came up with this grand idea? Lynn Margulis. And this was in around 1981. So like, it's kind of funny, I was 11. So this is probably like when I started studying cells. So mitochondria and chloroplasts were once free living bacteria. And they were engulfed, endocytosis, right? They were engulfed as perhaps food by ancestral eukaryotes. So they formed an endosymbiont, a partnership where both were going to benefit from the other structures. So this is showing you that a little bit. So the mitochondria, let's first of all go all the way back to the early days when we didn't have oxygen in the atmosphere. So bacteria didn't have a use for oxygen. So originally all of the early cells were anaerobic, right? And then at some point you start making some cyanobacteria that make oxygen from photosynthesis. Now we have oxygen available, and you know, there's just some genetic mutations that allow certain cells to do things that other cells couldn't do before. So now you have some cells that are actually able to utilize that oxygen being made. And so now you have aerobic bacteria. So the idea is that one cell engulfed one of those aerobic bacteria. Without digesting it, living symbiotically, that aerobic bacteria can make energy for the host cell. And the same idea happens with these chloroplasts. Certain cells were able to photosynthesize, like cyanobacteria, and then create food. So again, one cell engulfs another cell, and now they have a mutual relationship in that the inner cells are protected and by the outer cell, and then the inner cell is making food for the outer cell. So that's the idea on how the mitochondria and the chloroplast came to be inside of the cell. And then you can also talk about inner foldings of the outer membrane becoming the endoplasmic reticulum and the Golgi apparatus, and so on and so forth. Okay. So the plant cells, we know, have that central vacuole. I kind of already talked about it. We've talked about the animal cells with the food vacuoles. Contractile vacuoles, I have an animation of this I wish I would have put in there. They look like stars, and these are for protozoas, and you know, anything living in the water is going to take on water, right? So it needs a way of getting rid of the water so that it doesn't explode. So these act as a pump. So as it fills with water, and then it'll squeeze, contractile, squeezes all that excess water out, the contractile vacuole. And I feel like I already spent a little bit of time on that, so I'm going to speed up by continuing. Things that we might use vacuoles for storage of proteins and organic ions, things that we need for cellular reactions, the pigments that we're using for color, but they're attracting some form of light, so they're gaining energy in that sense. Defensive compounds against herbivores, so different excretions or secretions that they would use, and then they can let things in and out. So you guys already did a nice job of labeling the cell. A few of us left out nucleus because maybe we wanted to call the inner one the nucleus. So you know, you have a nucleus, and inside the nucleus you have a nucleolus. And then the picture that you were labeling before was pointing at the chromatin or chromosomes, the genetic material inside. So when I was going through it, that seemed to be the only one that was kind of common to miss. Your lab, which is what you're going to see in that video, has to do with surface to size ratio, surface area to volume ratio. You should be able to calculate this. The formula doesn't need to be memorized. It's on the formula sheet for AP Biology. So the big idea is that as cells get bigger, the volume increases, which makes them less efficient, right? Things need to get in the cell into the nucleus. So the bigger that cell becomes, the less efficient it becomes, which is why cells reproduce, so they can get smaller. So this is just talking about the greater the surface area to volume, because you have less to get through, the better. And that we kind of talked about with the enfoldings. And this has to do with the efficiency, amino acids, carbohydrates, proteins, different ions, ammonia that needs to get out. So things coming and going. The less space you have to get through here, the better. The more reaction area you have, the membrane itself, the better. So surface area is good, volume not so much. So the greater the surface area to volume ratio, the better. Okay, what questions do you have? I knew I was out of time, so I kind of started shortening things up there at the end because I talked so much at the beginning about announcements. Okay, so that for the most part was old news for you. So in the Google world, you're going to open up, I put this all as one, the lecture and the video and the endosymbiotic theory worksheet. So you can access all of those from that one site. And I want to stop sharing.